Good afternoon, everyone. This is, as you know, one of the highlights to the academic year when we actually recognize distinguished university professors. As you know, it's the highest recognition of our faculty. So I'm truly honored to be with you today. I know your friends and colleagues and some of your students of those who are being recognized. And this is a celebration of our most exceptional members of the faculty that are already full of lots of exceptional people. And this is to recognize extraordinary professors at the institution. You are scholars, you're educators, you are leaders. And I've had a chance to review some of your research, and it's absolutely fascinating and fundamentally important. I'm proud to be here with Lori McCauley, our provost and executive vice president for academic affairs, and Dean Solomon, who is doing a fantastic job as Dean and Vice Provost for Academic Affairs at the Rackham Graduate School. We are here to celebrate three people: Patricia Whitcup, Kevin Chung, and Deborah Lowenberg-Ball. These three singular members of our faculty are opening incredible new insights into evolutionary biology, repairing and rebuilding arms and hands, and redefining the practice of teaching in mathematics. I understand we're going to have a little bit of a test about our mathematical skills. Together, you are united by an eager and unstinting curiosity, a relentless pursuit of excellence and understanding, and a deep commitment to our students and our community. Together, you exemplify the ethos of our institution, our excellence and interdisciplinarity, and our calling, our mission, our values, and our vision. Together, you, along with so many others with you here today, are solving fundamental challenges, providing a life-changing education to our students, lifting our society, and transforming us into what we hope will be the defining public university. So we're so proud of you, everything that you've done in your fields, everything that you give to the University of Michigan. You really are an inspiration to us all. As I said, we are here to recognize you with the university's highest honor for our professors, extraordinary scholars, leading educators at the University of Michigan, to be distinguished university professors. So on behalf of the university, on behalf of the regents of the university, congratulations again. And now it's my privilege to invite Provost Lori McCauley to the podium to introduce our first lecture. Have fun, everyone.
Thank you, President Ono. As many university events as we have, there's too few that we get to be at together, and this is one of them, and really one of the best ones. Welcome to all of you. I'm really pleased to welcome faculty, staff, students, friends, family, and the public to this event today. And each year, I'm reminded that it is also one of the most consistently fascinating events on campus and truly a highlight of our academic year. Thank you to all who helped to make this possible, especially our event staff, our honored speakers, and Mike Solomon for your leadership in the event. And now it is my great pleasure to introduce Dr. Patricia J. Whitcup, the Deborah E. Goldberg Distinguished University Professor and the Arthur F. Thurnau Professor of Ecology and Evolutionary Biology and Molecular, Cellular, and Developmental Biology in the College of Literature, Science, and the Arts. Dr. Whitcup is a world-renowned evolutionary geneticist and a gifted teacher who has helped transform U of M's genetics and introductory biology courses. She's had a profound impact on her department at the university and within the scientific community. The major theme of her research is the experimental dissection of how the evolution of gene regulation processes themselves drive organismal and species-level differentiation. Along with the members of her laboratory, Professor Whitkop studies the genetic basis of trait differences within and between species, with an emphasis on the regulation of gene expression. She was selected as a John Simon Guggenheim Memorial Foundation Fellow, elected as a Fellow of the American Association for the Advancement of Science, and awarded the Friedrich Wilhelm Bessel Research Award by the Alexander von Humboldt Foundation. Her work centers on the origins of biodiversity. Over the last few decades, great progress has been made in identifying the specific genetic changes contributing to trait variation within and between species. Today, Dr. Whitkop will share some of the lessons learned from her team's research about how organisms develop and evolve. Dr. Whitkop, please.
Thank you very much. Thank you for that generous introduction. It truly is an honor to be here today speaking to you and to receive this recognition. I was an undergrad here at Michigan. I realized this morning that was 30 years ago, and I still remember being downstairs in the league that was a cafeteria at the time, having lunch with my husband, now husband, Keith Whitkop, who has always been my biggest support and cheerleader. I'm particularly honored today to have the opportunity to name this award after Deborah Goldberg, who is in the audience today. Deborah herself is a distinguished university professor and an Arthur F. Thurnau professor. She was a professor at U of M from 1983 until 2019, including chair of one of my two departments, EEB, from 2001 to 2013. She is a world-renowned population and community ecologist, as seen by her own distinguished university professorship, election to national societies, and a special issue of the Journal of Ecology being put together in her honor. She is also a dedicated educator who has literally written the textbook for her field. And the reason that I selected her, as great as those things are, is really she's a transformative leader, and she was, and her impact on the EEB department, on the College of LSA, and on the university as a whole, I think cannot be overstated. She has been a champion for making the academy more accessible and inclusive, including through actions such as starting the university's first Bridging Masters program, the EEB Frontiers program, and creating the MSci Academy, which was part of the MSTEM, again, sort of creating opportunity and supports for students. And then finally, the personal support that I received from Deborah, starting here as a new assistant professor during her term as chair, also was really profoundly impactful. So Deborah, thank you. I'm gonna try to not let the tears come. We can do that after together, so. Yeah, thank you. As Provost Macaulay mentioned, I'm a biologist and I work in this area. I'm often known for telling people that I have the best job in the world. I literally get paid to wonder how the world works and try to figure it out. For me, that wonder started with DNA. This is an incredible simple molecule. It's made up of 4 bases, which we refer to as A, T, G, and C. And the number and order and arrangement of those bases is what makes a cell either a bacteria or part of a tree or an earthworm or an elephant. And I still find that an incredible thing. So this DNA includes the instruction manual that controls development, makes an organism what it is and how it functions. It's also the molecule that's passed from one generation to the next, and with slight changes leads to and explains the process of evolution. So I put these two in a circle because to me that's how they are. Developmental systems are the product of evolution, and evolution happens in the context of existing developmental systems. So even though they're often studied in different departments on campus, and my work therefore sits between, for me it's kind of literally like a big circle of life, right? So by being at the intersection of these fields, I have the pleasure of working with colleagues in the Department of Ecology and Evolutionary Biology, in the Department of Molecular Cell and Developmental Biology, and also connected to the med school in bioinformatics. And as I said, our work really fits at the center of this, fundamentally trying to understand how changes in DNA sequence alter development and give rise to phenotypic differences we see. I am fortunate that I don't do this work alone. These are some of the undergrad, graduate, postdoc, staff scientists I've had the pleasure of working with over my nearly 20 years here as a faculty member. I wish I had the time to tell you all about each one. They're all incredible people. So I'll have to just shorthand it in the interest of time and say that they have not only made the work that we've done possible, they've made it better, and they've made it a lot more fun. So thank you to those of you that are here today. And I'll follow up with you individually and tell you my gratitude there. Okay, so a little bit about the science that we do. All multicellular organisms, including us humans, start as single cells. That cell divides to make 2 cells. They divide to make 4 cells, 8 cells, and so on. And each time the cells divide, they make a copy of their DNA so that the new cells have the same genetic material. As that process occurs, the organism is not just getting bigger from this cell division, it's also differentiating. So the cells are taking on different functions, different shapes, different activities. And they do this despite the fact they still have the same DNA, right? So I told you the DNA's instructions, and yet we can get all these different cell types. The way that happens is through a process known as gene regulation or gene expression. So even though the same DNA is in all the cells, and our DNA contains our genes, not all the genes are turned on or active in every cell. So this schematic, which is literally a slide taken from IntraBio, and I think I've used it in genetics too, shows how different sets of genes are on in different cell types, and that's what allows them to do different things from the same starting DNA. So we have genes 1, 2, and 3 happen to be expressed or turned on in kidney cells, genes 4, 5, and 6 in skin cells in this example. Okay, the information for how those genes are turned on, the information that tells when, where, and how much of a gene product to produce is also encoded in the DNA, along with the coding sequence for what makes the protein eventually. This schematic is actually a picture of a physical box that a former postdoc made to take to the Detroit Zoo to tell people about our research. And what it's showing is that here's the pointer. We have a region of DNA sequence that's the gene. It encodes a part of that encodes the protein product, which is the thing that actually works in the cell, as well as DNA sequences that control regulation or expression of that gene. When thinking about gene regulation in a variation or an evolution context, these components are important because if we mutate and disrupt the function of the main coding sequence of the gene, it's like turning off a light switch and that gene no longer works anywhere in the organism. However, we have these different regulatory regions that have their independent switches that control different parts of the body. So I could have labeled these sort of eyes, wings, abdomen of the fly. And when you mutate those, you can change expression only in a subset. And that's a powerful tool, if you will, for evolution. That you can cause a change in the eye pigmentation, but not the rest of the fly. Okay. This is a little bit more molecular detail, but still very cartoony, believe it or not, if you're not a biologist, to how this process works. So these non-coding sequences, these cis-regulatory regions or enhancers, bind proteins that are encoded by other genes, and they interact with the primary gene in a way that turns it on. Okay. And I share this level of detail only to point out 2 classes of changes that could occur to affect a gene's function. The first are cis-regulatory changes. We can change this DNA sequence itself, which will affect what binds to it, and therefore when and where the gene's active. Or we can have mutations in these proteins, which also can change the expression of the gene. Work from the whole field for 25 years, including some of my own, I'm going to give you the punchline first, has shown it's those regulatory regions that often underlie diversity. So this is actually an image from my own work, and I'll walk you through it briefly. I keep getting the wrong button instead of my pointer. 3 different species of fruit flies. Their wings have been taken off. They do have wings. We're just making it easier to see pigmentation. And they have different patterns and intensities of body color. The second panel here is a close-up of their abdomen, and this third panel is a close-up of a region of the developing abdomen, so earlier, actually during pupal development. So if you think about a butterfly and a chrysalis, that's the stage we're talking about before it's an adult fly. And what we can see when we look at where that gene is turned on, that is what's shown in yellow. So each of these squares have cells throughout them that all have the same DNA, but there's a subset that are producing this particular protein, which happens to be named yellow. And so you can see the species that make stripes have this gene in the stripes. The species that are darker have more of this protein than the others. So this observation suggests that changes in the evolution of this gene's activity contribute to the change in pigmentation. And we see the same thing in wings, right? So this seems to be general. Now, if you care less about fly pigmentation than I do, no judgment, I wanted to give an example to show that these principles that we're learning, changes in cis-regulatory regions, underlying variation, contributing to phenotypic variation, are of course not limited to flies or pigmentation. This is a video I'm not playing, it's just a screenshot of the YouTube. You can watch it on your own time if you'd like, describing work that has been shown that shows that differences among humans in lactose tolerance are caused by changes in a regulatory region. So babies around the world can digest lactose, lactose with their lactase protein. But in most populations, as they age, that protein gets turned off. The expression is stopped. And so the adults struggle to digest lactose. There's variation in this trait around the world. And in fact, the populations where lactose tolerance, or we call it persistence, the enzyme basically doesn't get turned off after childhood. It stays active to adulthood. Are highest, are most common in parts of the world where dairy farming sort of started. So you had natural selection pressure leading to basically the selection to favor this type of mutation that keeps the gene on. Okay, so back to fly pigmentation, and we also see variation geographically in this trait, and we can go and look and try to understand what might the selection pressures be. That's some of our work. We also study some pigmentation differences between flies that we don't think are tied to environmental factors, but rather mate choice, something called sexual selection. And I'll show you a brief video clip here. If you look very closely, there's this little spot here. That's actually the tip of a male fly's wing. So most male flies don't have these spots, but this species does. And I hope this is going to play when I hit the advancer. Yeah, you'll see him showing off. This is their courtship ritual for this species, and he sort of shows off his wing spots. The species that don't have the wing spots don't do that behavior. So it seems that there's a tight link between the two, and some of our work has sort of studied that and decoupled it in various ways. Okay, now all of this work has taught us that understanding gene expression is really important, and if we can understand that, hopefully we're stepping closer to understanding phenotypic diversity more generally. This motivated a new area of work in my lab that's just focusing on gene expression and looking at it across the genome, so at all of the 20,000 genes in, say, a fly's genome. I'm not going to get into the technical details, but I will give you a little insight in that we have our DNA sequence, and the way it makes these proteins I've been referring to is it first copies itself into RNA, and then that RNA is the template for the protein. We now study the RNA as sort of an intermediate. It's just more accessible here to see where those genes are turned on or active. Okay. Perhaps not surprisingly, as you compare RNA expression between different species that have been diverging for longer periods of time, it becomes more different. And so that's what this plot is showing. This is just, you can think this is like more and more expression divergence, this is less, this is increasing evolutionary time, right? And so we have data here from fruit flies, Drosophila, mammals, and yeast. And just to orient you, this closest mammal comparison is human-chimp. This one out here is humans and chicken. And so you can see we have comparisons in yeast and flies that go further than that. So one thing that we found striking about this was not just that they become more different over time, but how similar that rate of divergence is. And this suggests to us that there are some shared features that all eukaryotes have about how their genes are regulated, that was known, but about how they evolve. Okay? All right. One of the other contributions that I've made in our work is to figure out how we look at those expression differences and figure out where the genetic changes in the genome that cause them are located. As I told you before, they can be in the cis-regulatory sequences or these transacting factors that connect them. And I'm not going to, again, go to the details, but we found a strategy for disentangling that that's now used not just among evolutionary geneticists, but human geneticists, developmental biologists, kind of widely in the community. And what that work has shown is that as the evolutionary time increases, more and more of those differences are due to changes in the cis-regulatory sequences. Okay. Once again, to link this out to human variation, which some of you, for unknown reasons, may care more about, there is a lot of variation from human to human in gene expression in a given tissue. When we look and map the genetic basis of that, it tends to be in the same cis-regulatory regions as well. So I'm not, again, just going to skip the technical details, but that's the punchline. So even though we study this in flies and pigmentation and yeast, the principles we're learning apply broadly. Okay, and the final bit that I'll just mention, not in any much detail, is one of the newer lines of research in the lab, although we've been doing it for a while now, and that is asking the question of why do we see the patterns of variation that we see, right? So we now have the tools to measure and describe this, but why, right? Why do we see that type of variation? And I think of this as the possible and the actual, which is actually the name of a scientist, Francois Jacob, that I literally named my son Jacob after. We have mutations, right, are the primary source of variation in all living things. And so that's kind of the space of possibility. I don't know where I am on time, but if I have time I do have time for a brief tangent. A few years ago, I put together a museum exhibit at the Museum of Natural History, and I decided to focus on this question, the sort of input of mutations, and I highlighted it with an eye-catching image of lasers shooting out of a cat's eyes, right? And it was like, why haven't we evolved cats that shoot lasers out of their eyes? Is it not advantageous? Cats are predators? Surely that would be great. The reality is, though, that there are no mutations that can occur in a cat's genome that will make lasers shoot out of its eyes, right? And so that inability from a mutation standpoint limits the potential of evolution and how living things can evolve. So we study the effects of new mutations as they arise, specifically the effects of new mutations on gene expression. Over the passage of time, and I've clarified generations here, some new mutations get passed on and others don't. Sometimes this is because the new mutation makes the individual more likely to reproduce. That gives them what we define as higher fitness. The other time, it's by chance. So if a student walking to class on a rainy day steps on an earthworm in a puddle, it doesn't matter if that earthworm had the best DNA in the world for earthworms, it's dead, and it might not reproduce then, right? And so there's both chance things that happen, and there is the sort of role of natural selection where someone or something is more fit. Okay, so we study the mutational input to understand what's possible. We look at the variation within a species, between species, to see what's actual, like what's actually happened. And I sort of think about this as studying not only the survival of the fittest, which is a phrase probably most of you have heard before, but also the arrival of the fittest, right? You have to have the mutations that create that variation before selection can act on it. And to just give you a little insight into how we do this, we sort of start, this work is done in yeast, I'll mention that. It's really hard to screen through fly genomes and find small changes in expression, but yeast are single cells, and the GIF that you see playing there is actually the budding and the division of those cells. The yellowness comes from a fluorescent protein that we use to monitor the expression of a particular gene. And so by doing this work in yeast, we can have this population of literally millions of cells. These cells divide every 90 minutes. This is sped up, but it's still a very quick process. We can introduce mutations. We can use technology, flow cytometry, for those of you in the know, to isolate individual cells that have changes in their expression that are introduced by the mutations we entered randomly. By studying that, we can then understand the possible. Again, these would be examples of distributions of effects of those new mutations. And we can compare it to the actual. So we can go to natural populations, look at the variation that's there in the DNA sequence, recreate that sequence in the lab, measure its effects, and ask, is it consistent with a random sampling of mutations, or is there some bias to it? So for instance, here we have two distributions of sort of, again, actual sort of polymorphisms in the real world, mutations for what could happen, and they don't look different. So we would say this looks like variation that may be due only to chance. Here they do look different, and we may say natural selection seems to have favored mutations that increase this trait's value or disfavored the ones that decrease it. So by characterizing the effects of mutations and comparing it to what's in the natural world, we're learning about the relative roles of neutral, that's the kind of drift step on a worm process, and natural selection, right, which is what leads to adaptation. Okay. So putting this together, what have we learned in the scientific realm, or sort of in the biology realm? DNA sequences controlling gene expression are an important source of phenotypic variation. That is true everywhere we look. Bacteria, again, to trees, to elephants. It's true within species, it's true between species, it's true for all kinds of different traits. It's a really important part. Okay, we've also learned specifically that changes in these cis-regulatory regions, the ones that are the switches controlling gene expression, matter a lot. And the further apart you compare over evolutionary time, the more they contribute to that. Okay? So that seems to be a hotspot or a major contributor to evolution. And then finally, both natural selection and mutational biases, which again would be, this is that circle that I was talking about of new mutations arise in the context of an existing developmental system, right? So that can shape what new phenotypes are possible. That both of those processes play a role. Okay, and if you're not a Drosophila evolutionary geneticist or even an evolutionary biologist more broadly, why should we care about these things? I think there's a number of reasons. First of all is just straight up fundamental science, right? Curiosity driven. We are curious about how the world works around us, how living things are what they are, how they came to be, and to me that's enough, I'll say that, like to drive this work. Specifically, however, it does help us understand these general principles about how genetic variation relates to trait variation. Again, that's the main thrust of human genetics too. We understand how DNA sequence controls development by this work, and we understand how evolution works, sort of at a practical level. More generally, thinking about helping, say, solve world challenges or problems, understanding how genetic variation arises, how it's passed from one generation to the next, and how these can spread through space and time was really critical for understanding how the SARS-CoV-2, right, the virus that caused COVID, spread throughout the world. You all hear about the different variants and how you need to get a booster because the strain is evolving, right? These are all based on evolutionary principles and the type of work we do in the lab. Understanding the evolution of antibiotic resistance, right? Antibiotics play such a key role in our medical sort of healthcare today that the evolution of resistance to that is a really scary thing to think about. Agricultural bioengineering, many of these, we figure out how to make things better, sometimes by experimentally evolving them in controlled settings, so that's another application. And finally, it's no secret that the climate is changing around us, and if we can understand how living things might be able to evolve, how they're likely to evolve, we may be able to put measures in place to help us prepare for that change and protect from some of its scariest impacts. So that's where I'll stop. I'm happy to answer any questions you have. Thank you all again for your attention, for coming, and to the people in the audience in my lab, thank you for all your contributions to this work.
Thanks very much for your lecture. I'm Mike Solomon. I have the pleasure of moderating the questions. We have microphones, and so if you have a question to raise, please raise your hand or indicate otherwise, and we'll bring a microphone over to you. And we have a question here to begin. President Ono.
So I've done similar kinds of research with human genes and identified polymorphisms in cis-regulatory elements and also in transacting factors. In yeast, would you say that if you look at evolution, that it's usually a polymorphism in a cis-regulatory element that's responsible for it? Because, so I'll give you the two examples. There's something called Bair lymphocyte syndrome in humans where it's a transcription factor, where there's a mutation in the transcription factor that turns off the gene, which results in the absence of HLA antigens at the surface of lymphocytes, making you immunodeficient, right? But similar kinds of mutations in the cis element have less profound effects on the phenotype relative to the functioning of the protein. So why would you think, if you compare what we and others see in humans and you see in yeast, there's a bias towards polymorphism in cis elements in your case, and representation of both transcription factors and cis elements in humans?
Yep, great question. There's a couple answers. One, we see this not just in yeast, we also see that pattern of increasing cis in flies and where the data exists in mice. So it does seem to be a generalizable trend. The second thing is I would say that the types of disease mutations that you're talking about are not the stuff of evolution, right? These are the ones that cause really bad things, which is why we study and care about them in the human context, sort of human medicine context. And so in fact, the way you phrase the question is actually part of the answer. Because transacting mutations do tend to have more deleterious effects. In fact, we published a paper in Science last year kind of directly teasing apart and measuring these types of effects. That's why we believe the cis tends to be favored. So yeah, the answer is sort of in your question. Thank you.
Well, that was a wonderful technical question. I'll ask the next question, which was naturally needs to be about cats and laser beams. So I understand that you use that as a way to draw in the young folks into your natural history. I'm just really appreciated the shout out to the Natural History Museum. Can you talk a little bit about, there's a distance between the cats and laser beams and your research. And once I can see how the kids came over with that, and how did you bridge the next step to get them into where they are seeing your actual science?
Yeah, so my goal in that drawing them in, and what I hope to achieve in the museum exhibit, was really getting them to think about that second piece of evolution. Too often, in my opinion, evolution is about survival of the fittest. And these caricatures of, I won't get on my soapbox about that common depiction of a linearity to evolution, which is not true, and there's lots of bad things implied by that that are misrepresented and misused. But I wanted them to think about that first mutational step of something has to actually be possible. There has to be a mutation that can introduce this change for it to then be selected for and favored, right? Evolution doesn't have a magic wand. It can't, it's also not anthropomorphic, right? So I think the hope was to draw them in and get them thinking about, okay, not everything in this world around us is optimized for what it can be. And B, that is in part because of our understanding of biology, and again, this mutational process, which is happening in all living things all the time, right? That we can't evolve just anything we can imagine, right? That there is this constraint and that barrier that has to be overcome before something can become adaptive and selected for. The exhibit also included, at the time I was putting it together, my son was in 2nd grade, and I asked him and his classmates to draw an animal they could imagine that they'd never seen. And they created some wonderful stuff, and that ended up in the exhibit too. So just trying to separate reality and sort of the X-Men sort of view of the world.
It's wonderful.
Yeah. And I really just said X-Men in my Distinguished University Professorship talk.
We have time for one more question. We have time for one more question from the audience. I'm going to ask just a quick follow-up then. So I understood then that that it's not so much the gene that provides the function, it's actually the regulation, the thing that's regulating it, that is the thing that matters in the long run.
Yeah.
Is there a conjecture as to why that would be as opposed to the opposite?
Yeah. So I would say they both matter. Neither can work alone. In fact, I think I've started a paper with alone a coding sequence is inert. Like the DNA sequence by itself, if it's not expressed, if it is not transcribed into RNA, translated into protein, it can't do anything in the cell. So it isn't that the coding sequences aren't important. It is that when mutations occur in them, they're more likely to be deleterious, as you said, and so they tend to be selected against. So they tend to be highly conserved over evolutionary time. In fact, if I go back and actually before I was really a practicing scientist, around the 1980s, it was believed that elephants had elephant genes and mice had mice genes and tree had tree genes. And one of the big radical realizations or observations in biology in that time period was as we started sequencing genomes and mapping gene functions, we learned that actually these things have remarkably similar sets of genes, but what they do with them is different and how they do it is different, and that's all about the regulation. So to give one quick, hopefully quick example of that, one of the first case studies that demonstrated this was with an eyeless protein. So in flies, genes are named for their mutant phenotype. So if you have a mutation and the eye doesn't develop, we call it eyeless. Very creative, right? The yellow gene is named yellow because when you break it, black pigment isn't produced and the fly looks yellow. So this eyeless gene, however, was found to be very similar to a human gene that causes various eye diseases and disorders. And there was an experiment done in Walter Gering's lab where they took the human copy of this gene and put it into a fly that had a broken copy of this gene, and it restored the eye's function. So basically that human protein worked in the fly, and it didn't make a human eye, it made a fly eye. And that's because it's one of these transcription factors you just heard us talking about, and in humans and flies, they regulate different things. So the high order switches are conserved, but what they do is changing. And that's, again, that regulation piece.
Great. So, well, please join me in thanking our distinguished university professor for her lecture.
What a great start, and you can see why she is both a distinguished university professor and a Thurnau, because she takes incredibly elegant science and makes it really digestible to a broad audience, so thank you. I now have the honor of introducing Dr. Kevin C. Chung, the William C. Grab Distinguished University Professor of Surgery and the Charles B.G. Denancreed Professor of Surgery, Plastic Surgery, and Orthopedic Surgery in Michigan Medicine. Dr. Chung is an expert at repairing and rebuilding hands and arms due to congenital abnormalities, trauma, or destructive arthritic conditions. He has trained hundreds of college and medical students, residents, fellows, and faculty at the University of Michigan Medical School, and has taught surgical techniques to doctors caring for children in the world's poorest countries. Dr. Chung is the Chief of Hand Surgery for Michigan Medicine and the Associate Director of Global Reach, the medical school's global health program. He has served for 12 years as the Assistant Dean for Faculty Affairs, a role in which he's responsible for tenure and promotions. He's a distinguished visiting professor at Tsinghua University. Dr. Chung was the 75th president of the American Society for Surgery of the Hand and past president of the Plastic Surgery Foundation. He has published over 850 peer-reviewed manuscripts and has over 41,000 citations with an H-index of 93. He's published over 500 book chapters and 40 textbooks. He's had continuous funding from the National Institutes of Health for more than 25 years. He's the editor of Hand Clinics, past deputy editor of the Journal of Hand Surgery, both American and European, and he is the editor-in-chief of Plastic and Reconstructive Surgery. Wow. He's going to talk to us this evening about enhancing the lives of countless patients over the course of his 30-year career at the university. Dr. Chung.
Well, thank you, Provost Macaulay. That's so generous of you. Mark Twain said, to succeed in life, you need two things. You need ignorance, and you need confidence. When I came to Michigan over 30 years ago, I was ignorant how difficult it is to succeed in an academic institution as prestigious as us. However, I was very confident that the institution would support us. The leadership would support us. Our team members will support us, and most importantly, our patients will support us. And I received this award on behalf of everyone who was part of this fantastic journey for 30 years at University of Michigan. As an academic surgeon, you need to do three things. Well, the clinical part did not come late. A few months after I came to Michigan, I got a call. Dr. Chung, we're airlifting a patient whose scalp was pulled off. I go, what? scalp was pulled off. She has long hair. Her hair got caught in the machine. And we look at the literature and say, how many of these cases have been done? Not many, 1 or 2. And we struggled to go to the operating room. We went to the operating room. Blood was everywhere. And this poor 37-year-old lady has a scalp come off. I cannot show you the pictures because it was censored. However, However, we spent 8 hours hooking up every single vessel, the artery, the veins, and so forth. And it was 8, 9 hours continuous under the microscope. 3 sets of residents were cut off because they passed out during the surgery just because of fatigue. And at the 8th hour, when the artery continued to perfuse the scalp becomes thicker and thicker, but all the veins are hooked up and unable to drain. I was thinking to myself, I was so tired. I was thinking to myself, dear God, please help me and my patient. Miraculous thing happened. We took a vein for the leg. We found a vein for the scalp. We tunneled it all the way down to your external jugular, which is high flow. And after hook it up, the scalp starting to go down. And we know we have a chance. And this is a patient with a few weeks after surgery. You can see the hair is growing. I cannot close the scalp just because the scalp was so edematous. And this is her, and this is her, and this is her, like 6 months later. We all have imposter syndrome. We all feel that we come to an institution like this, why did they pick us? Well, after this case, I think the imposter syndrome has gone away. I know we can do magnificent things at Michigan to help patients who have no means, who has no other chances to go anywhere, and we can help them. Professor Monahan, a British surgeon, said the perfect surgeon must have the heart of a lion, the hands of a lady, and not the claw of a lion, and the heart of a sheep. And throughout our entire career, my colleagues and I have done miraculous things, such as a patient with a hand cut off and was censured, but that's okay. And we're able to put the hand back, and through the dedication of our therapists, many of them sitting here and also on the web, they recovered the 17-year-old's hand, and he's able to go back to active lifestyle. And of course, we treat many different complex injuries. This is serratus fracture that women 65 years and older has about 10% chance of getting this kind of fracture from osteoporosis. And what about this one? The most artistic and miraculous surgery that we can do in surgery. This child was born without a thumb. You can see that thumb is rudimentary. What was the picture to the right? That picture is an index finger that is made into a thumb. Can you tell it's an index finger? No. The artistry of the surgeon must be in full display in taking care of these complex problems. Charles Darwin said it's not the strongest species that survive, nor the most intelligent, but rather the ones most responsive to change. I did not know how to do research. I was trained as a surgeon in general surgery, in plastic surgery, in hand surgery. I was going back to my hometown in Atlanta to be a community surgeon because I can operate. I have not written a single paper. I do not know how to do research. However, amazingly, my colleague, my chief, asked me, Kevin, apply to the Robert Johnson Foundation program. I said, what is that? Just apply. Well, in the middle of my hand fellowship, I got a call. I was operating. Dr. Chung, the president of the Robert Johnson Foundation from Princeton, want to talk to you? I said, what? So I went out, and then the president said, Dr. Chung, congratulations. In the 40 years history of Robert Johnson Foundation, we picked the first surgeon, and that's you. My career changed entirely. I am no longer going back to my hometown in Atlanta, Georgia. And this person is so influential to me because he founded the Johnson & Johnson Company, and General Robert Wood Johnson founded the Robert Wood Johnson Foundation that created a cadre of the best health policymakers that shape the US healthcare system today. We are building not only the framework of stone and steel, but frameworks of idea and ideal. And I was the first cohort at University of Michigan, in which I'm so indebted to my teachers. Many of them are listening on the web, who has helped me, a surgeon, the first surgeon ever in the program, and who doesn't know how to do anything. I have to learn, you don't have to be great to start, but you do have to start to be great. And I started from grounds up. I know nothing about research, we attend seminars, we do research projects. And what are these things? Study design, data management, sampling, a heavy dose of statistics. I don't even know what p-value is. So as a surgeon going through the program, it's just daunting. And however, Thomas Edison said, keep on the lookout for the novel ideas that others have used successfully. Your idea just have the original only in its adaptation to the problem you're working on. And every project I've done failed because no one knows what to do with a surgeon, and we want to measure outcomes. We want to know whether our surgical procedure worked. You know what? The only thing that worked during those 2 years is the Michigan Hand Outcomes Questionnaire. Michigan Hand Outcomes Questionnaire that we developed in Michigan has submitted in hundreds of papers, 50 different conditions, cited numerous times. It's considered a legacy questionnaire because it has withstood the test of time. It's been translated to every single language in the world. And you see the side, world domination? Indeed, it is world domination, the Michigan questionnaire. But now, what do I do with that questionnaire? Who is this? Renoir. He was born in France, 1841. He suffered from rheumatoid arthritis, a disease that attacks all your joints. Your neck is attacked, your spine is attacked, you are decrepit, your hands are deformed, you cannot walk. 1% of the US population has rheumatoid arthritis. And Renoir came from a poor family. His father sent him to be an apprentice to make porcelain. But he wanted to be a painter. And he was roommate with Monet, actually. And Monet is vivacious. He is so charming. People invite him for feasts regularly. Whereas Renoir, he's quiet. And I read his biography. At one time, for a whole month, the only thing he ate was potatoes. And Renoir was in this amazing period in which Impressionist movement happened. And the painting was to represent the first impression, this ethereal, magical, and dreamy-like pictures that we all love to see Impressionist painting. And Renoir said, art should be pretty. Yes, pretty. Life brings enough unpleasantness. Why not approach it from the light side once in a while? And he was cheerful. You see his paintings, all bright, beautiful. The babies are just so gorgeous. Everyone's gorgeous in his painting. And he's such a so-called motivated figure in Impressionist art. However, life dealt him a bad card. He got rheumatoid arthritis in the midst of his most productive time when he's 50 years old. His hands are deformed. He cannot walk. He's like so thin because malnourished. And I was able to find Renoir's hand in 1903, in which you can see the deformed hand. But he continued to paint these things that you see in museums. How did that happen? And this is a picture of him with his wife, in which he's in a wheelchair. His family took care of him, moving around to do things so they can continue his passion. And there's a misconception in the language about impairment and disability. And I used that to write a paper about his adaptive mechanism. Impairment means when you lose a body part, you are impaired. Does that mean you're disabled? No, it doesn't mean you're disabled. And what does that mean? This is a patient, a farmer, a Michigan farmer who came to see me. His hand was cut off in a machine injury in his family farm. And he goes, Dr. Chung, I cannot drive a tractor. Can you give me a thumb? I'll figure it out. I'll make you a thumb. What is that? That's a thumb? It's actually a second toe. I took his second toe and make him a thumb. Now he can grip his wheel. He can drive. Now he become greedy. Dr. Chung, can you give me some fingers? It took me a long time to make you a thumb. You want some fingers? Okay, I'll figure it out. So I took 2 toes on his other foot, and I make him 3 fingers. And he's able to use his hand to do things. He sent me pictures that he's able to tie his shoes and stuff, but that's not the only thing. He showed me a picture that is fixing his tractor at home. He is impaired, but he's not disabled. And the coping mechanism that Renoir uses is that he put cotton between his fingers, cloth, so he doesn't get ulcers because his hands are deformed. And this is just an amazing picture. In the sense that he's so cold that he asked the cat, kitty, kitty, come over here, sit on my lap. And cats keep him warm. And the curators later on are able to use cat hair to verify that those are original Renoir painting. How about that, right? And his coping mechanism, another picture I found, that he's able to put his palette and he's able to drive around and paint paintings. And so he has mechanisms. However, critics are continuing to say that Renoir is not the same Renoir. He's unable to paint big structures. He does small things. And towards the later of his life, he painted this for no reason. He painted a picture of his son as a huntsman in 1910. And I read the biography from his son. His son says, I don't even like hunting. I have no idea why my father painted this. I'm 6 feet tall. And people hypothesize that Renoir painted this because you say I cannot get my hand to paint something. I'm going to paint my son 6 feet tall to paint this one. The indomitable spirit of the master. The triumph of the human spirit is undeniably wonderful. The pain passes, but the beauty remains. Now I know where the Michigan questionnaire should go to. The Michigan questionnaire, for the first time, has an aesthetic component. Surgeons for many years kind of say, you know what, just get the hand working. Beauty, aesthetics has no role in our reconstructive armamentarium. But when I look at you, I see two things. I see your face and I see your hand. And beauty, normality, is what human beings aspire. This is a man with rheumatoid arthritis. You can see his hands are crooked. And he hides his hand behind his back when he takes pictures. People, no matter who you are, want to be pretty. We did the first study that defined the outcomes of joint replacement for rheumatoid arthritis patients, a 3-center study funded by the NIH from the most prestigious centers in the world, University of Michigan Hand Center, Curtis National Hand Center, and Palmertaft Infirmary in Derby, England. And this is what we found. 5 years after surgery, you can see the left hand looks pretty normal. The right hand is still crooked. And patients valued this. And now we have a consensus between surgeons and rheumatologists and say, when a patient has deformities of the hand, send it to a good surgeon, and preferably to University of Michigan. And this is what this patient is. And then we start ambitious without going crazy. Life isn't about being perfect. It's about being so ridiculously passionate that people think you're a little crazy. And my team members here will verify that Dr. Chang is pathologically insane. It's not a little. And Cochrane report, which is a consensus panel around the world, said despite 200 years of treating this fracture, this serratus fracture that I showed you earlier that happened in a lot of women with osteoporosis, there is no evidence to support any of the treatments we prescribe for this injury. Is that a strong statement? 200 years, surgeons just flip a coin and prescribe surgery for you. And we want to do the most ambitious study possible. 24 centers, 100 investigators, countless support staff to run this study. We do not know what we don't know. This study should take 5 years. It took us over 10 years when we published no papers, just because it's so hard to randomize patients. But this is a most ambitious study in musculoskeletal diseases funded by 2 institutes for NIH. And success is walking from failure to failure with no loss of enthusiasm. We want to quit so many times, but the entire team stayed for 10 years. And we are able to solve the puzzle. And even though we publish a lot of papers in JAMA, in surgical journals and orthopedic journals, this, I think, is the most influential editorial that I wrote for JAMA Surgery. I told them the fallacy that we put for 200 years is that when we look at patients who are 65 years old, we think that, oh, no. We need to look at the physiologic age. A person who's 70 years old, who's still working, playing golf and stuff, he needs to be treated like a 40-year-old. You cannot treat everyone based on age. You have to look at the physiology of the patient. And that fundamentally changed how we treat this serratus fracture. We take the patient as a personalized medicine approach rather than a statistics. Education. We profess to profess. We are teachers in this academic center. And I study Chinese literature, and this is a classic treatise called Sun Zi Bin Fa, the Art of War by Sun Zi, who is a mystical general from 200 to 400 BC. And Sun Zi, the interesting about him is that nobody knows who he is. But during that warring period, the big states swallow the smaller states, and people rely on the strategies to be able to survive. And this is one of the principles in Sun Zi Bin Fa. 士卒如爱子，故可与俱死, which means that literally that you treat your team as your beloved sons because they will follow you to the deep valleys of existence. And I'm so blessed. I've trained hundreds of students in my career. Many of them are here because they value what we value at M-Choir, which is a lab we have. We want to conduct research to advocate those without means and speak for those without a voice. And I firmly believe that mediocre teacher tells, the good teacher explains, the superior teacher demonstrates, the great teacher inspires. And we want to inspire a whole generation of surgeons who can serve the unfortunate. And we don't want to be a compliant culture because we do it because we have to. We want a committed culture. We do it because it is who we are. And the Japanese have worked out the meaning of life. I can tell you that. There's a word called ikigai. Iki means live, gai means shield. And there's 10 principles. And what does ikigai have to do with this? In this reflective moment when I make this presentation, is that I've been all over the world with my team to teach hand surgery because many children do not have the ability to live without a functioning hand or feet. And this is a picture in Nepal. We find the meaning of life, content, and fulfillment with me taking care of patients in Central America, Honduras, and teaching hand surgery in Vietnam. We thrive to help those who are disadvantaged. Carl Sagan wants us to have humility. And you know what Carl Sagan is? He's this famous American astronomer who makes science popular. He was looking for extraterrestrial life. And humility is not thinking less of yourself, it's thinking of yourself less. And this is a beautiful book that he wrote. He took a picture, the picture was Voyager 1, they take a picture of the Earth. Let's show you that picture. This is us. And Carl Sagan said, look again at that dot. That's here, that's home, that's us. On it, everyone you love, everyone you know, everyone you ever heard of, every human being who ever was, live out their lives. And I feel so strongly that as a surgeon and physician, trust is not a guarantee, it's something you earn. The trust in the surgeon as a person, the trust in the surgeon's skills and training, the trust in the science that underwrite those skills. And I'm going to end this presentation by showing you one of my kids. This child was born with constriction band syndrome. And many hospitals turn her down because they don't know how to take care of it. Constriction band syndrome is amniotic fluid, amniotic band wrapped around your extremities, and you can't tight enough, cut off circulation to your feet, to your hands. You wind up with no hands. And this child was sent to University of Michigan, in which we did reconstructive surgery to release the constriction band. 7 years after surgery, 9 years after surgery, she has a functioning hand, and she's able to write and go to school. Amazingly, just recently, a few years ago, Dr. Chung, my name is Selena. I'm 17 years old, one of your patients several years ago. It affected both my hands and my feet. After meeting many doctors, they found you. They're so happy that you can help my situation. I recently found you on the internet through UN website. I want to thank you for the work you did to help me. 3 surgeries and 16 years later, I'm able to live and function like a normal person could. Thank you for the effort you put in. Now, this is one of my kids. Once a man was asked, what is the meaning of life? He replied, life itself has no meaning. Life is an opportunity to create meaning. As an academic surgeon at University of Michigan, I have lived my ikigai. And I am so grateful that I have the opportunity to share our collective life story to you. I'm most appreciative of the support of everyone in this institution during my 30-year career. Thank you so much.
Thank you so much. We're open for questions. Professor Ona will begin.
So I love music, and your hands are very important to be a musician. There's a condition that's affected a lot of musicians called focal dystonia, as you know, which is a neurologic condition. One treatment method is with Botox, right? But I'm curious, as a hand surgeon, because what results ultimately is a curling of the fingers, and the inability to move them, which you have to do to be a pianist. And so, as a hand surgeon, is there some other way other than Botox treatment to treat focal dystonia?
Focal dystonia is a really complex problem because it's multi-origin. It could be I mean, we don't want to say psychological, however, the brain and hand are not connected. And we know a famous pianist who can only use one hand to play. And my family is sitting back there. My wife is a pianist as well. So we are very, very attuned to musician problem. And I think that when our therapists are also very in tune with the musician problem, when you see a musician coming in, you have to give the musician a holistic approach because they're high-performing people. They cannot treat them like any other person because the kind of tasks required to play the instruments are so unique, very similar to a surgeon, which means that the practice of surgery on musicians is absolutely intricate. We spend a lot of time with them, we guide them, we talk to them, we need to know what's affecting them. One of the things that musicians does is that they do crazy stuff. The musician can climb up the roof and fell off the roof. And we treat a bunch of musicians who break their hand by shoving snow from the roof. And so musicians, just like everybody else, have common ailments, but in terms of the function of the hand, they're very unique. So you're absolutely right, Santa.
Questions? I'd like to offer one. I am just tremendously in awe of your spirit, the spirit of your patients, and I'm connecting that with what sounded like a major intervention, which was the creation of a survey, a hand questionnaire. Can you just talk a little bit about how something like that seems so regular, how that has had the impact that you showed in your talk? Like how did you realize that was what the need was, and even what are you asking in that questionnaire?
Absolutely. 30 years ago, the outcomes movement started. That's why I was sent as a Robert Johnson Scholar, because we want to know what patients feel rather than what the surgeons prescribe. We want to know how the hand will work in the totality of one's existence, which means that the Michigan Hand Questionnaire measures not just how the patient feel the hand can move, but also the pain perception, also the ability to conduct daily activities. But one of the most amazing things is that the ability to feel normal again, which is a aesthetic component. I think that for the first time in surgery, people realize that looking normal, looking nice, reconstruction is just as important as our function. And the editorial written by the editor of Journal of Hand Surgery many years ago was that we need to stop thinking of just function. We need to pay attention to how we build the hand to make it pleasant to look at and also pleasant in society. And that's a fundamental difference. So I think the Michigan questionnaire is unique because no other questionnaire has that kind of consideration in the holistic portion of a patient. Yeah, so we're very proud to be able to put the Michigan stamp on this questionnaire.
Thank you. I think we'll close the questioning here, but I just want to say 2 of our lecturers have now mentioned cats in their talk, so the pressure is on for the 3rd speaker. Congratulations, Professor.
Wow, thank you for a truly inspirational presentation and a very inspirational career. I'm delighted now to introduce our final lecturer for the evening. Deborah Lowenberg-Ball is the Jessie Jean Story Fry Distinguished University Professor of Education and Arthur F. Thurnau Professor, the William H. Payne Collegiate Professor of Education in the Marcel Family School of Education, and a research professor in the Survey Research Center in our Institute for Social Research. Professor Ball is an internationally respected scholar whose work is grounded in the study of mathematics education and teaching practice. Her pioneering research has shaped teacher education, providing important insights into the ways that inequity and oppression regularly pervade normative teaching practice and how teacher education and development can disrupt this. Professor Lowenberg-Ball began her career as an elementary teacher and continues to teach young children every summer while making visible, for research purposes, the day-to-day work of teachers. She earned her PhD in curriculum, teaching, and educational policy with a minor in mathematics education at Michigan State University. She's worked with mathematicians, teachers, and mathematics educators to identify the specialized mathematical knowledge needed for teaching and showed its relationship to and the difference from disciplinary mathematical knowledge. This work has become the basis for her receipt of the International Commission on Mathematics Instruction's Felix Klein Medal for Lifetime Achievement in Mathematics Education Research. She is the most highly cited author in the world in that field. Presidents Barack Obama and Joe Biden appointed her to the National Science Board. She also served as our Dean of the School of Education for 11 years. Professor Lowenberg-Ball's lecture concerns a potentially intimidating question. Do you know mathematics well enough to teach fourth grade? She will discuss her work focusing on the study of teaching practice and its entailments, particularly for teaching across difference and for redressing pervasive harm. Professor Ball.
Thank you, Provost Macaulay. How is the sound? Is it good? Okay. So the title of my lecture, as Provost Macaulay just said, is, do you know mathematics well enough to teach fourth grade? So how many people think you do know mathematics well enough to teach fourth grade? Okay, so by the end of this lecture, you'll be able to answer that question for sure. I'm going to start with a simple example. So imagine that you're teaching fourth graders, and you're teaching them to multiply multi-digit numbers. So let's start with what you might actually be teaching. So let's say if you were going to teach a problem like this, obviously you would have to know how to do this problem yourself. So take a moment and just, whatever you can do to figure out the answer to that, just do it really quickly. What's the answer? Come on. What's the answer to 49 times 25? 1,225. So undoubtedly, you actually can multiply these two numbers. But the point is that when you're teaching fourth grade, that's really only the beginning. Obviously, you have to know what it is that you'd be teaching. But that doesn't really get you very far, because when children are first learning to multiply, they aren't necessarily going to get the answer 1,225 at the beginning, because it's a complex algorithm. So I'm going to show you now 3 answers that children can reasonably come up with when they're first learning to multiply multi-digit numbers. And I'm going to give you a short amount of time to see if you can figure out what mathematical steps produced each of these answers. So I'm just going to give you a short amount of time. You can talk with somebody nearby. Okay, that's about all the time you get, because if you were actually teaching fourth grade, it's not like you can sit around and sort of ponder this or go down the hall and talk to people. You actually have to be quite fluent at this. So let me just get a little read of the room. How many of you were able to figure out all 3 of them? Raise your hand. Just look around. There are no hands up in this room. How many people could figure out 2 of them? 1 person. How many people could figure out 1 of them? Okay, quite a few more. So part of the point here is that knowing the mathematics yourself is crucial, but it isn't actually what it turns out to be crucial in actually teaching. So I'll explain only one of them right now to you, and I'll leave the rest to you. So let's look at A, okay? So with A, imagine that you multiply 9 times 5 equals 45, right? And normally you would probably call it carrying. You would put the 4 above the 4 in the 40. So imagine that you add the 4 to the 4, and that gives you 8. And then you multiply 8 times 5, that gives you 40. And similarly, on the second one, 9 times 2 is 18. You write down the 8 and carry 1. But if you add the 1 to the 4, then you have 5, and 5 times 2 is 10. Then you get 108. Now, I could ask you to explain why you don't add the carry in first. That's another whole question you would need to be able to explain. I'm not going to ask you that right now. But that is a completely reasonable thing to do when you're first learning to multiply multi-digit numbers. I won't explain the other two, but I could do that during the Q&A if you like. So let me just take a step back for a moment. In our society, it's commonplace for adults, I'm sure you've heard this, for people to say, I'm not a math person. Not only is it commonplace, but it's basically considered to be quite acceptable to say something like that, despite the fact that many important career pathways depend on quantitative reasoning and mathematical knowledge. And in fact, in everyday life, there are many cases where one needs to be able to reason quantitatively and use math, or to think critically about quantitative information. So it's actually not something we should be laughing off. It's actually something we shouldn't be taking as normal. So I became a teacher after college. I think Provost Macaulay referred to this. And I will say that it was the hardest thing I'd ever done. College looked easy compared to teaching elementary school. But I was fortunate because I had a remarkable mentor. And this is the person for whom I chose to name my Distinguished University Professorship. Dr. Jessie Jean Story Fry, who I know is here. Can you just at least raise your hand? She was my principal for my very first year of teaching and all the years that followed. I'm thrilled to be able to recognize her for the profound role that she has played for me in all of my work. Here's a picture of the two of us in my second year of teaching. You can see her on the left and me on the right, and these are my second graders. Here we are a couple of months ago when we met to talk about my naming of the Distinguished Professorship in her honor. An experienced teacher herself, Jessie embodied a deep commitment to the success of young students, especially children of color, and the many children that we had in our school who spoke languages other than English at home. To me, she was skillful, she was tough, she was demanding, and she was deeply caring as she nurtured me as a beginning teacher. As a beginning teacher, I didn't actually begin by focusing on math at all. I was really preoccupied with helping my students learn to read, to write, to use language. I was learning about their cultural resources. I was learning about their languages, about their families, and thinking about how to use all of that to help them grow. But after a few years, I found myself very puzzled because helping my young students learn math turned out to be really complicated and difficult. I found that they were already not identifying with math, not learning math, not enjoying it, and I had not been my experience with the other subjects I had to teach, like science or history. I could do the math myself, of course, as we sort of saw already, but teaching it was another thing entirely, and that was the genesis of what turned into one of the main lines of research that I've been pursuing across my career. It seemed to me that if one wanted to reverse the pattern we see in our society that marginalizes whole groups of students, people of color, women, and all these people who grow up to say that they're not math people, it seemed to me that teaching mattered critically. I thought it mattered for disrupting those patterns. I thought that it mattered to understand better what that work of teaching was. The work of teaching is actually a very broad field that I and my colleagues have been engaged in. And so I chose just one aspect of that research to show you today, based on the research that I've done with many colleagues over many years. And that one aspect that I chose for today is what I call the mathematical work of teaching. With my research team, we did something somewhat unusual. We turned the problem upside down, which is instead of trying to pontificate or predict what it is that teachers would have to know, which many people do all the time and say things like teachers need to have this many courses or they need to have a degree in mathematics. None of that was really rooted in what the demands of the work were. And so we decided to study teaching practice itself and see whether we could enumerate the kinds of challenges that come up mathematically in the course of the work. What comes up for teachers as they're trying to teach mathematics? What tasks is it that they have to do? And what mathematical understanding or reasoning is involved in carrying out that work? So at the beginning of this talk, I engaged you in a very simple example of that by showing you the difference, I hope, between knowing how to multiply on your own, yourself, and being able to understand multiplication well enough to be able to figure out what might a child be thinking when they begin to learn something. There are lots of other tasks beyond the one I showed you. For example, trying to figure out what students have written when they've written out a solution to a math problem, trying to pose a really strategic mathematical question, trying to interpret what a student has said, using diagrams or representations to explain a mathematical concept, and many, many more things like that. So many tasks of teaching that sound kind of generic turn out to be filled with mathematical demands. And my team and I called this specialized mathematical knowledge. Specialized in the sense that it was a way of knowing mathematics that was attuned to the demands of the work. And we built a kind of theoretical model out of the empirical research we were doing. And all I really want to say about this right now is that on the left-hand side of the diagram, you see subject matter knowledge, in this case mathematics, and you see something called common content knowledge. That is the portion of mathematical knowledge that teachers need that is just like what anyone who uses mathematics needs, like to be able to multiply, for example, or to be able to add fractions, or to be able to figure out the solution to an equation. What was most interesting to us was the portion of content knowledge that we called specialized content knowledge. The right-hand side of the diagram is also interesting, but it's not what I'm talking about today. The right-hand side is more amalgamated knowledge, including things that you would expect, like knowledge of how students learn, or typical misconceptions, or how to teach a particular idea. Those are all mixtures of knowledge. On the left-hand side of the diagram is still mathematics. And in order to validate our hypothesis that this was actually mathematics and not some kind of pedagogy, we built test items. This was a very interesting part of the work. We attempted to measure the nature of this kind of knowledge and did multiple kinds of validation studies that I won't be able to talk about today, but they were fascinating. And one of the most important outcomes of that research and those validation studies is that we were able to establish that that knowledge was specialized, specifically because often mathematicians couldn't answer questions that were purely mathematical, and you would expect that they could answer, but they couldn't. We also learned that teachers who scored better on these items were more able to improve their student learning than teachers who scored less well. So that was all very important. I said I wouldn't talk about it. I'm not going to talk more about it. But I did feel a kind of tension with this work, even though this was all good news. The tension I felt was that by building test items, it felt as though we had reduced what I was interested in to something very static: knowledge, test items. And my experience of teaching and my study of teaching suggested that that's not static work. It's dynamic, it's listening, it's interactive. So something felt a little off. So now I want to describe another branch of this research that has helped us to advance this theory. So over all of these years, I have continued to teach children long after I was a full-time teacher. And this teaching that I've been doing has been a kind of design research in which I've been trying to develop ways to engage young children in significant mathematical work that to broaden the kinds of mathematical experiences that they have in school, to learn what young children are capable of doing mathematically, and to confront deep patterns of inequity that have pushed out students of color, girls, and others of historically marginalized groups. This branch of work is something I have come to call first-person research, because what I'm doing is studying the work of teaching from the inside. You can imagine doing that with other kinds of professional practice. It afforded a view of the nature of this work of teaching that complemented our studies of other people's teaching in other classrooms. So now, in the rest of this talk, I'm going to take you inside of that work, that first-person research, with a brief visit to my third-grade classroom on one particular day. At the beginning of the class on this day that I'm going to show you, I was ready to return to work on even and odd numbers that we had been doing for a few days. And before I started the lesson for that day, I asked my class whether anyone had comments about a meeting that we had had with fourth graders the day before. I won't go into what that meeting was about, but we had had a meeting. And a boy named Shay, you see him here at the board, raised his hand. I'm going to play you what he said in response to my question, whether anyone had comments. You won't see him at this point of the video, but you will hear him. So listen to what he says in response to my question.
More comments about the meeting? I'd really like to hear from as many people as possible. What comments you had or reactions you had to being in that meeting yesterday. Shay?
I don't have anything about the meeting yesterday, but I was just thinking about 6. I was thinking that it's an odd. It can be an odd number too, because there could be 2, 4, 6, 2, 3 2s. That would make 6, and 2 3s. It could be an odd and an even number.
I had never heard this before. What was Shay saying? 6 can be even and it could be odd, both? But the other children objected, some of them pretty vigorously. Listen to Tina, for example.
People's comments? Tina?
I disagreed with when he said that 6 can be an odd number. I think 6 can be an odd number because, look.
Jillian Bernadette?
6 can be an odd number because this is even, odd, even, odd, even, odd, even. How could it be an odd number? Because even, odd, even, odd, even, odd. Because 0's not odd.
So Tina is using the number line to build her argument. A few more children after this also questioned Shay, and a boy named Kip, who's sitting near the board there, asked Shay, prove it to us that 6 can be both even and odd. So Shay does that by drawing 6 circles on the board, and he shows that there are 3 2s, but there are also 2 3s. And that's what he uses to try to establish his argument. But many children are still not convinced by this. And at this point, a girl named Lynn asks to go up to the board. So listen to what she does.
Oh, 6 is an odd number. Why don't we? Let's see. 5, 6, 5. Okay. Let's say 10. 1, 2. And here are 10 circles. Then you would split them. Let's say I wanted to split them by 2. So 1, 2. And look, 1, 2, 3, 4, 5. Then why do you not call 10 an odd number and an even number? Or why don't you call other numbers and odd numbers and an even number?
I didn't think of that way. Thank you for bringing it up. So I say it's 5, 10 can be an odd and an even.
Yeah, but what about other numbers? Like if you keep on going on like that and you say that other numbers are odd even, maybe we'll end it up with all numbers are odd and even. Then it won't make sense that all numbers should be odd and even, because if all numbers were odd and even, we wouldn't be even having this discussion.
It would be.
So obviously this doesn't end up in the way that Lynn anticipated. Instead of countering his argument, Shay just accepts her addition and accommodates her counter-argument. So what is going on so far from a sort of, when you think about the work of teaching and the work of teaching, mathematical work of teaching, there are actually many things going on. It's important who's talking, whose ideas are being centered. A white boy has initiated this discussion with his unexpected idea, and he's been challenged by several children of color, including several girls. The work of teaching involves a lot of power and a lot of discretion, and the moves that a teacher makes have consequences for who is seen as smart, what it's understood to be mathematics, and what's considered to be valuable mathematical thinking, and that is all at the teacher's discretion in the way the teacher moves. Hence, a clue into this problem of the mathematical work and the judgment involved in teaching. So I want to focus on the specialized mathematical knowing that's involved so far in what you've seen. Shea is suggesting that 6 can be considered both even and odd, and this could be heard as simply wrong, and ultimately the children should just be told that 6 is even. What about Tina? What is she saying? And what about Lynn? What's the mathematics involved in what each of these children has now contributed to this discussion? So what Lynn does is she accommodates what Shea did. She also groups by 2. And she says, it doesn't matter how many groups it is, an even number can be divided into groups of 2, and it doesn't matter how many groups it is. So one thing is to see here is that Shea is relying on a definition of even numbers that's based on groups of 2, and so is Lynn. Tina, on the other hand, is using a recursive definition, looking at the number line to show that even and odd numbers alternate on the number line, starting with 0 as even. So seeing and thinking about what these children are talking about and its mathematical significance is central to the work of teaching. It's not just about naming 6 as even, which actually the children already knew. They already knew that 6 was even in the sense that if you named the number 6 and said, is it odd or even, they would say even. But they didn't have any definition for it, which meant that if you showed them some other large or arbitrary number, they wouldn't necessarily have a way of determining whether it was even or odd. You need a definition for that. So the specialized mathematical knowing matters for hearing the mathematical significance of this and the opportunity for creating space for children to be learning mathematical practice and to show up as smart. So at this point, from what you've seen so far, one more child wants to speak. Ogechi. So listen to this and hear what Ogechi is doing.
Wait, I didn't really need to show you.
You don't need to show something? What are you trying to say?
I just wanted to say if you wanted an odd number, you just had to take one off it.
Why would that work?
Because usually odd numbers are like
Tina, can you hear
this.
Ogechi okay?
That it's kind of like have ones in the middle, like 5. And there's a 1 in the middle, or 9. I think are you circles? This 2 together, together, and this 2 together, and just 1 left. And even numbers like 6. You can't get anything in the middle. There isn't one.
So interestingly, if I asked you to work on trying to name what the mathematics is that Ogechi's doing, it would take a little bit of time. So I'm going to try to show you what she's done. She's actually offered 3 mathematically equivalent definitions of odd numbers. So the first one she offers is, if you want an odd number, you can just take 1 off of it. So you have an even number, you take 1 off, you have an even number left, you have an odd number left. The second thing she says is odd numbers have 1s in the middle. So here she's working with dividing a number into 2 equal whole number groups and showing that if you do that, you have 1 left in the middle. That's equivalent to the first one, but it's not the same statement. And third, she says, going over more to Shea's looking at groups of 2, odd numbers have 2s in them also, but they also have 1 left. So these are 3 mathematically equivalent definitions that would be perhaps worth the children trying to establish what makes these the same, and are they the same or different from Lynn's, from Shea's, from Tina's. So what do we see at this point about the mathematical work of teaching? First, I want to say that the conscious effort to disrupt patterns of marginalization rooted in racism, sexism, and linguicism depends not only on commitments to justice and serious understanding of the way those patterns have repeated themselves in our classrooms in this country, but it also depends fundamentally on knowing mathematics. The children who speak up and challenge Shea and challenge his assertion are all girls of color. And their brilliance involves not only believing in that brilliance, but hearing it, and hearing what they're saying and understanding the depth of their mathematical thinking. Ogechi, for example, I don't know if you noticed this, initially demurred to go to the board. She said, I don't need to show anything. And it was only when the teacher said, what were you trying to show? She turned around and went back up to the board. And her contribution was enormous for the class's work. A commitment to advancing equity, I argue, is hollow without the capacity to hear the sophistication of the children's mathematics and to recognize its importance. But it's not just seeing and hearing. Teaching isn't just seeing and hearing children. Knowing and using mathematics for teaching is fundamental to being able to create the spaces that are big enough, like we just saw, for children to show up and engage in authentic mathematical work. It's also necessary for building on what they bring up to bring this to a kind of mathematical growth for them and to help them flourish, to see themselves as mathematical, and to position them as math people, not math people. And if all you can do when you're teaching third grade is re-explain that we call 6 even, the opportunity to work on those goals is lost. And the opportunity to support children's flourishing is similarly lost. So why does this matter? Many reasons. I'll just name a couple. One central reason this matters is because of the harm that we know obtains in classrooms when good mathematical thinking is misconstrued as error, which easily could have happened here. Another part of what makes this important is the importance of positioning children and their mathematical work as authentic mathematics and building on that thinking. But a third, and one I think not to be dismissed, maybe in particular when we think about where we are as a nation, is the opportunity for mathematical work to promote habits of civil discourse. Lynn starts her contribution with this statement that I didn't play for you before. Before she goes to the board to show the 10 circles, this is what Lynn does.
I think what he is saying is that it's almost, I think what he's saying is that you have 3 groups of 2, and 3 is an odd number, so 6 can be an odd number and be even number.
Do other people agree with that? No. Is that what you're saying, Shay?
Yeah.
So notice that although she disagrees, and we saw that, she begins by actually listening carefully to what her peer is saying. Certainly appreciating the power and possibilities of the work of teaching mathematics and supporting it could change a country where so many exit school not identifying with math, saying that they're not good at math, and thereby excluded from pathways that require that kind of mathematical thinking. It could disrupt racialized and gendered patterns of who profits from our current systems of mathematics education. But also more broadly, it could contribute to a country where people are able to listen and disagree with respect and regard for differences. Before I close, I want to acknowledge that this work has been funded generously over many years by the National Science Foundation and supported by the many colleagues with whom I've been privileged to work and to learn. This is a very incomplete list, but I hope I've done well at naming quite a few of the crucial people who have helped me learn to do this work. Other teachers, graduate students, educators, and people from fields as various as mathematics, psychometrics, policy studies, philosophy, and critical studies. So I'll close with this question. Do you know mathematics well enough to teach 4th grade? Thank you.
Thank you for that lecture. Questions for our distinguished university professor?
Thank you, Deborah. That was awesome. I love the peer learning aspect of it. I'm curious just to bring forward one of the quotes from Dr. Chung's lecture around survival requires adaptability, right? The benefit of that. And so I'm curious your thoughts of how we've evolved teaching and how potential impacts like generative AI may have on teaching 4th graders.
How we support teaching?
How generative AI may change.
This is the AI question.
Right.
I think that's a very interesting question. I'm thinking of writing a piece called Have We Lost Our Minds? Because The thing that's very interesting to me is what are the aspects of teaching practice that could be very, at this point, from what we understand, really supported by artificial intelligence? And what are the pathways to follow right now that probably aren't the places to go? What are the dimensions that AI right now doesn't have that capacity? And I think that's a really important question. So I see a lot of people trying to build tools using AI. Some of them don't really make sense. I've tried feeding in some of these videos to see if ChatGPT can suggest what to do, and the answers are always completely ridiculous. However, I think there are things that could be of great assistance to the complex work of teaching, and that's where I think the question lies, is like, how can we take advantage of the power in ways that suit the nature of this work? Its ethical and contingent demands may not be the places to look first.
You mentioned to me when we sat together that you knew of my brother, Ken, who is a number theorist, and he was also an assistant producer of a film about Ramanujan called The Man Who Knew Infinity. And I was very moved by your remarks and the videos, because as you know, the core of that story is that a great British mathematician, Hardy, received a crumpled up letter from India, from Ramanujan. And he said, this person I have to meet because he's either a genius or he's crazy. We now know that he was a genius. And it wasn't because he had the finest teaching, because he lived in a house with a dirt floor in India. And he, as you know, went there on a scholarship and became the youngest mathematician to ever become a member of the Royal Society. And his work is still the foundation in questions that are inspiring mathematicians today. And even today, people say, he must be crazy because we don't understand what he's asking. But his impact has been enormous. So here's the question. What makes them special, some people like that? In your teaching, in those classrooms, can you identify an extra Ramanujan?
So you were wondering about what it looks like to see really unusually gifted humans at a young age, right? It's an interesting question. I've often thought about that because gifted education is kind of it's a thing among families, and in particular kinds of communities, everybody thinks that their child is gifted, and they're often, what I'm interested in is how gifted so many young people are, and how little space and stage we give them to do that. I think we would have more people we would consider to be very talented in math if we didn't close in the mathematical space the way we do in school. It's really remarkable how narrow the mathematical diet is that children get in school. We shouldn't be that surprised when they hate it by the time they leave. However, you are asking an interesting question, and I would say across my many decades of teaching, I've probably only encountered a small handful of children who were just completely different from other children I had ever met, who were doing things you would never expect 5- and 6-year-olds to do. And that does seem different to me, but that's not the commonplace, and that's not the thing that preoccupies me. It's the loss of all the children who are actually extremely capable mathematically and who get turned off by teaching that isn't well supported and by a lack of opportunity to really play with the nature of mathematical work that these children were having. Thank you for that question.
In the front here, Carla? There's a microphone coming too.
Deborah, you mentioned a range of knowledge or knowledges that teachers need to have in order to facilitate mathematics instruction. Which do you think is hardest for us to cultivate among beginning teachers, and accordingly, what is the best way of cultivating that hard thing?
Of all the kinds of knowledge, yes. I think that's so what Carla is asking is, of all the things that teachers might need to know or be able to think about and reason about, what might be the hardest to cultivate? And of those, what can we do to cultivate this? I think it's a very important question because teachers, unlike any other profession, whether it's surgeons or people who work in biology, people who become teachers grow up in our schools, and they've already been spending years of watching teaching and developed many ideas about what learning is, what learning requires, what mathematics is, all of these things. And so our work when we're training teachers requires confronting things they already think they know. That isn't true in a lot of other fields. Imagine training pharmacists, for example. They don't spend all their time in pharmacies, and they don't come in with a great number of preconceptions. So the ways that children have learned in school about who's smart and what it is to be smart or what learning looks like have to be, in some sense, confronted. And we're still learning about what that takes to do that really, because they can learn to say the right things about why it's important to value all children or why it's important to understand the gifts that children have, but their behavior and their habits may not change along with their beliefs and their knowledge. So it's not just a knowledge question, because teaching is a practice. And so finding ways to confront habits that, we all teach in everyday life. You all teach, whether you teach professionally or not. So helping people become teachers who can change what the opportunities are that children have requires a kind of undoing as well as a kind of building. So that isn't a direct answer. I think actually almost everything we do requires understanding that they're not blank slates when they come, and they have habits that have to be undone. But probably the hardest thing of all isn't content knowledge. It's probably actually learning to see children and understanding the capability of children whom they've learned from school aren't capable. Black children, girls, children who don't speak English, they come with those, and that's very hard to undo. That doesn't mean we shouldn't be doing it, but it's hard, and it can be very easily performative instead of deep.
Thank you so much for that lecture. Let's join me in thanking our distinguished university professors.
Thank you.
As we close the session, two things. Specific mention to our DUPs to please not leave right away. We're going to take a picture of you. And for those that have joined us today, please stay for further engagement and the reception that's going to begin right now at the back of the room. Join me in thanking our three lecturers for their wonderful presentations today. Thank you so much.